back to the afternoon session of the Modeling Immunity Workshop at Fields Institute. I hope all of you managed to catch a good break and lunch in the last 30 minutes. And I hope that you guys also enjoyed the talks and the panel discussion this morning. So following that, our next talk, we'll switch gear a little, and we would like to hear a little about modeling immunity to malaria from Dr. Lauren Charles. So please help me welcome our next speaker, Dr. Lauren Charles.
Great, thank you very much. Thank you. And thank you to the organizers for giving me the opportunity to talk. You'll notice that my talk's a little bit maybe different in flavor than what some of those great talks from this morning are talking about, both in topic, but also in sort of the scope of the work that I'm going to talk about today. So to give you a little bit of an outline, I'm going to give a very brief background into malaria and then talk about 3 different projects that I've been working on, one that's completed, it's a little bit older, and then sort of a couple of things that have been built more recently out of that. This is work I've been interested in for a while, but sort of got put on hold for the last year and a half, 2 years based on sort of other things that have been going on. So to begin with, obviously, malaria is still a really major public health problem around the world, even though cases and deaths have been declining. In the past 2 decades, there's still, over 200 million cases a year. That's about the equivalent of half of the US population getting malaria, and more than 50% of the world lives in areas where malaria is endemic, with children under the age of 5 really accounting for most of those deaths. And so a couple things to know about malaria if you're not familiar with it. It's a 2-host life cycle. It requires the Anopheles mosquito and then a vertebrate host. I'll be focusing on the human here today in this talk. And there are multiple different parasite species, including 5 different parasite species that infect humans. But the focal parasite species that I'll be thinking about today is Plasmodium falciparum, which is the species that causes the most morbidity and mortality. And so it has this complicated 2-host life cycle over here in the mosquito, as well as in the human host, including sort of this repeated growth cycle and a transmission cycle. I'll try not to use too much of the jargon. There are a lot of sort of complicated biological words on here, but this idea that both hosts are required, and then there's sort of complicated life cycle dynamics in each of those hosts. So the other thing that's really interesting is there's a lot of heterogeneity in malaria prevalence. And this varies by all sorts of different things related to age, related to sort of spatial distribution, which is primarily seems to be related to environmental factors, maybe related to mosquitoes and the dynamics there, as well as through over the course of time due to seasonality and behavior. And we do see the acquisition of natural immunity, but not natural immunity against infection, but rather against disease. And this seems to appear after repeated exposure. And so I'm really interested in understanding how to model prevalence, malaria prevalence and malaria incidence, including all of these factors. But in order to do that, to begin with, there's a complicated process of acquiring immunity within individual human hosts. And so I'll start there. We know from a really nice set of historic data that there's variable course and duration of infections. So the 2 graphs I have here are 2 different patients. And you'll notice from the very different scales on the axes in the left and the right that the individual on the left had a very short infection, but with very high density over here within an individual host, whereas this patient on the right has, a much longer, on the order of sort of half a year here, infection, but sort of slightly lower order of magnitude, lower density. And so we see this split somewhat between acute, severe, high density infections and these chronic, maybe slightly lower density infections. In addition to that, and not sort of coming out of this direct data set, but from other work that people have done, that many of these infections actually harbor, some of these long infections and even short ones can harbor multiple genotypes. And when I say genotypes, I mean sort of multiple different aspects of the same parasite strain. Remember, we have multiple species. So I'm not talking about co-infection between different species, but rather some sort of genetic difference that accounts for maybe a slightly different phenotype, which I'll talk about. Right. And well, one of the things that we see, especially that we think is important in these longer chronic infections, is the antigenic variation of particular proteins across a single within host infection. And so we know there's this protein denoted by PFEMP1, Plasmodium falciparum, or erythrocyte membrane protein 1, that has a bunch of different forms, around 60 in any individual parasite, sort of represented over here with the domains in the colors idea. And the parasites only present one of those, only express one of those genes and present the protein associated with it on the outside of the red blood cell that it's infected. And then we see these shifts of which types are being presented over the course of an infection, which I've sort of drawn as cartoons here with these different colors and shapes over the course of this individual infection. This is, a schematic and not exactly, this was not determined from this particular data, but this idea that we see these different waves that are happening due to the antigenic variation of individual proteins being expressed at a phenotypic level by parasites within an individual infection. And so what's the role of antigenically varying these proteins? Well, the proteins sit on the exterior of the red blood cell, which the parasite has infected, and it allows for that red blood cell to adhere to other cell types. So in this case, we see it adhering to other red blood cells, to endothelium, and to immune cells here, which allows to avoid the clearance of the parasite, particularly when it binds in things like the capillary walls. But these types vary through that course of infection, and we kind of see something that looks like a synchronized switching of type, which means that there have a somewhat synchronized switching of exposure to the immune response. And so we're interested in understanding how the immune interactions, the immune system interacts with these varying proteins and leads to protection over the course of time within an individual infection, right? And so we did this by modeling a number of different aspects of the immune response. In this particular model, we included 4 aspects. One on the innate side, where we just considered it as protection against parasites that are there, and it was dependent on the total level of parasitemia in the infection. And then we included 3 different adaptive types that sort of varied based on the level of immune cells of those types. And we did it, we broke it off into 3 components. One where we considered variant specific, so it would only react to a particular variant that was being expressed on the red blood cell that the parasite infected. And then we considered cross-reactive subtypes. And so the variant specific being over here, this like particular blue circle being the protein, so it's reacting to the color and the shape. And then these cross-reactive subsets that were reacting to sort of one feature of that protein, maybe in this case, the shape that we have. And we also considered something that we called a general adaptive immunity, which was sort of a level of immunity to other proteins that the parasite is expressing on the surface of the red blood cell that sort of built up over the course of infection that we would see, sort of equivalent to degree days in some temperature-driven models that are out there. And so this led to a very large system of delay difference equations. Sort of in the blue here, I'm highlighting the parasite-related models, and we modeled each of those 60 plus variants individually within an individual host. And then we had sort of our 4 types of immunity there, where we broke those apart into, responses to the variant specific and the cross-reactive that we see there. And because it takes time for the adaptive immune response to develop, it led to this delay difference equation model that we're looking at. And I will note that we chose a difference equation model over a differential equation model in this context because the parasite has a particular life cycle where it bursts out of the red blood cells about every 48 hours. So we wanted to account for that sort of fixed delay between entrance into that cell type and exit into that cell type. And so with that, we were able to use this model to really look at, features that led to different components of these interactions. And so loosely, we saw that the innate immunity really drove the timing and height of that initial peak that we were looking at. And then the cross-reactive and variant-specific immunities came together sort of in a complicated way to really tell the length of these sort of oscillations that occur later through time. So we have these very sharp up and down oscillations that are related to the bursting of the red blood cells every 48 hours with the new parasite cycles. And then these longer ones that seem to be associated with different courses of different presentation of those antigenically varying immune cells. And then our general adaptive was really what caused this slow decay over time that led to these decrease in the overall peak heights through these chronic infections. And we found that the variant-specific and cross-reactive immunity were really what was important for ultimately driving these courses of infection to be lost and fall below that detection limit and stay below the detection limit. In this model, our model is a deterministic model, but we used a lot of stochastic variation in our parameters to understand what sorts of heterogeneities drove different types of infections. So on the host side, we varied the effectiveness of all of our different immune responses in different ways, sort of how effective they were at driving down parasitemia, how quickly the response activated, and how quickly or the rate of growth and decay of our immune cells, which was particular for our variant-specific and cross-reactive. And then on the parasite side, we varied a lot of properties related to this antigenic variation, including the growth rate of each of the variants, how the variants sort of moved between the different types, the rate at which we saw that switching, and sort of the cross-reactivity that we saw between different variant types. And with that, we were able to come up with some ordering of the role of different heterogeneities and understanding what's really driving these courses of infection, in particular, the difference between the acute and chronic infections that we have. And what we saw is that the immune properties were more important at driving what these effects were, meaning that variation, the prediction being that the variation in the individual patients would be more important than what was different in individual parasite types that we were working with. But if you sort of took our model and fixed the individual per se, and then varied the parasite within it, we still see a lot of importance of those parasite parameters. They're just sort of a secondary effect that we are working with here. And here, of course, because our parameters are varied across different ranges, I've scaled them on this plot to think about the range of interest that we're looking at for each of our parameters, and sort of that range being biologically determined that we were looking at. And so we see that this, our innate immune response was sort of the most important, but then we see differences for our cross-reactive and general adaptive and variant-specific sort of at certain outcomes. And then when we, on the right-hand side, if we fix our individual patient, we see this importance of our switching network and our variant-specific thresholds and cross-reactive thresholds coming in. And we did a lot more with this model. We actually considered co-infection in the context of two different sort of strains of the same species coming in together that may have some overlap in these proteins or may not have overlap in these proteins and may come in at different times. And there's a lot more that I could say about that, but really the sort of the driving home point is that our different immune aspects here are really leading to complicated dynamics that within the host of the complicated dynamics of the parasite within the individual human host that can lead to short infections and chronic infections. And in fact, when we see this co-infection between the two different types of the same parasite, then we often get complications in non-intuitive ways, things that when you add, say, two individually long infections together, getting a shorter infection, or one that does not transmit as well. So what I've been interested in is going beyond the context of an individual, but thinking about how can we think about immunity at sort of a more population level, but including immunity feedback. And so what I've talked about was published a number of years ago, but the next two components that I'll talk about are sort of newer and ongoing work that is happening. And so I brought up this idea that there's a lot of heterogeneity related to age, related to the environment, related to time. And so these pictures here, I'm showing our age-dependent distributions of parasite prevalence and different areas that have, say, different altitude or different levels of rain. And one of the things that we see is that when we have low transmission regions, we see a very flat profile. And in higher transmission regions, we see a higher peak among young children in terms of malaria prevalence. And so that's one of the things that we're hoping to replicate with this model. And so what we're doing is building an age-dependent model that incorporates immunity feedback. We have our human populations where we're considering both asymptomatic infection, meaning there's harboring of parasites, as well as disease, because we know there's built up a protection to disease, but not necessarily to infection here. And to simplify it a little bit, we consider our mosquito population to be in a quasi-equilibrium state that could be time dependent. So we have the ability to incorporate seasonality of mosquito populations, as well as our biting rates. Our interaction between our mosquito population and our human population are allowed to fluctuate over time that we're working with. So that if we have very large mosquito populations, we can sort of limit the number of bites that we have. In addition to that, we have equations that really track what we consider to be maternal immunity and acquired immunity here. And here we've considered thinking about the difference between clinical immunity, which would block against disease, and anti-parasite immunity, which blocks against infection here. And we're focusing more on the clinical immunity side. And what this means is that we have our transitions are actually functions of the level of immunity that we measure or track across the whole population. So we have this feedback of between the population level of immunity that is tracked by age onto the likelihood that we are going to move between different disease states in the human population that we're working with. And so it leads to this complicated PDE structured model here, where we have our age-dependent classes for all of our human compartments at the top, our ODEs for our mosquito model, which we assume to be in this quasi-equilibrium, and then these PDE again for our tracking of our maternal and acquired immunity. And again, this is ongoing work, but one of the things that we've been looking at so far is thinking about the variation based on environment and age. And so if we look in areas with low EIR, which is equivalent to few infectious mosquito bites per human versus high with many infectious mosquito bites, we see sort of different patterns in the immunity level here, the y-axis being sort of a scaled version of immunity that we're looking at. We always see a peak at very young ages, less than 1, due to the level of maternal immunity those individuals are acquiring, and then sort of a dip for our younger children, and then something that either flattens out for low EIR or one that's going to rise and ultimately lead to more protection as those children age as we move forward. And so we're interested in using this model that we're in the process of calibrating right now on thinking a lot about how immunity profiles at the population level are going to change with all sorts of different known variabilities added in, such as interventions, vaccines, bed nets, drugs that can alter the dynamics, or mosquito variability, or even human behavior. And I do want to highlight that this project has been really great. It's been done with a group of up-and-coming mathematical biologists through AMS Math Research Community project or workshop that happened last summer after being delayed from 2020. All of our meetings with this group have been virtual. I've highlighted the 6 individuals that were in this particular group. The other individuals worked on other projects in mathematical biology. And I want to particularly highlight the 3 research on the top, Li-Hong Zhao, Dennis Patterson, and Joan Puntz, because they're all postdocs right now. And so they'll be on the market, and they're really great. It's been a fantastic group to work with, and we'll have some more coming out as a preprint soon, hopefully. And then the third component that I want to work with is ultimately trying to tackle this idea of within host immunity at the individual level and the feedback at the population level, and sort of bring those loops back together. And so one of the things that we've focused on here is thinking about how parasite diversity really changes over the course of the infection cycle, and how that leads to sort of different levels of, say, evasion of the immune response or persistence of the parasite, both in individual hosts, but also at a population level. And this can incorporate changes in things like increases or decreases in infectivity or severity of disease, and lead to loss of effectiveness of both our drugs and our vaccines. And of course, it's going to be complicated by the idea of multiple parasites, multiple types of parasites occurring in the same host, which is something that we thought about a bit in that first project I talked about. And so this idea that we really want to incorporate dynamics both within our human host, but also within the mosquito vector, which is another area that I've spent a lot of time on thinking about parasite dynamics in that mosquito vector, although they are not so much immune modulated. So I didn't want to talk about them here today. And in this multi-scale model that Olivia Prosper and I have been working on, Olivia is also in that group with the mathematics research community, sort of working on that immunity feedback model as well. And we have a lot of variation at each of our three sort of scales within our human, within our mosquito, and at the between host level, and thinking about different variations that we have here. And so this idea of if we have some level of parasites, then we actually have this disconnect at within the humans between the transmission stage level and the total parasite level there, and thinking about how that alters the probability of transmission. And then we've done some work within the mosquito that thinks about what level of parasite do you enter in, and how that leads to the time between when the mosquito becomes infected and when it's able to transmit the infection. We see we've shown that there's been sort of a U-shaped curve there, as well as this idea of, using this idea of that difference in timing that, when you bite, when a mosquito bites in the parasite's life cycle within itself, it really matters about the probability of transmission, and in particular, which of these genotypes are being passed on. So if there is multiple sort of different strains in the individual mosquito, what's the likelihood that they're getting passed on? And we see that that's really important for understanding this idea of diversity, maintenance, and generation in here. And so one of the things that we see is like different factors play different roles in terms of thinking about the richness or the number of unique sequences that are getting passed around in these populations. There's obviously some known correlations about, higher probability of infection is going to, at both the mosquito to human interface and the human to mosquito interface, are going to lead in increases, but we see very large differences in sort of the scale of increases over here on the right-hand side that we see. As well as we know that a higher mosquito to human ratio is going to increase that diversity, but sort of what of those levels that we're working with. And we're really trying to think about how best to calibrate the interaction of our different processes here. And so one of the things that we see is that diversity is maintained for much longer periods of time when we have these much higher probabilities of infection, as well as shorter times to infectiousness. And that seems to be highly sensitive to this idea of transmission investment, which has been a really big question in malaria research about the propensity of the cycling within the human host to lead to more parasite growth versus transmission investment there. And so I've sort of given a really quick run through of a number of projects. I just want to highlight that one of the things that we see is we see very non-intuitive outcomes when we include immunity within a human host. And we're really interested in how those outcomes and the immunity profiles at a population level are going to feed back into one of them. And so we have this idea that the parasite diversity and the parasite sort of population and prevalence are going to be really sensitive to what's happening at this within host level, and the need to really connect these different levels of modeling together to really fully understand the dynamics that we have. And so I just want to end by acknowledging the people that I've worked with on the different projects I talked about here. So the work on the within human host modeling of malaria done with Carolyn Buckey at the Harvard T.H. Chan School of Health. A lot of the other modeling also has been done with Olivia Prosper, who's at the University of Tennessee, and then that immunity feedback being part of this mathematics research community group that's ongoing and has been really great experience. And of course, acknowledge some of my funding. And with that, I'd be happy to take any questions. Thank you.
Thank you, Lauren. So the floor is open for questions. You can type it in the chat box. I am keeping watch on the chat box, or you can unmute yourself and ask a question as well. So while waiting for questions from the floor, I probably have a question myself. Coming from Southeast Asia, when I was a child, I was affected by malaria as well. What sort of data that you use to fit your model? Do you look at the countries that are right now, having this endemic issues, or you're looking at historical data, back 50 years?
Yeah, so most of what I've done has looked at either historical data or, in fact, some in controlled infections, and not always with Plasmodium falciparum, but sometimes with, say, rodent malarias, that sort of thing, to get that data has been more on the side of what's happening in the mosquito, just because that's a lot harder to do in the field. But a lot of the sort of within human host data that I have worked with has been these much older infection studies, mostly because we know that they are single infection studies that we're working with, or we know we can select for only looking at single infection studies. There's lots of interesting data out there. The sort of field data that I have taken a look at and worked with has been primarily in sub-Saharan Africa, due to the fact that there really isn't co-infection or a significant co-infection between multiple parasite strains. And so it does allow us to focus more on Plasmodium falciparum versus sort of co-infection between Plasmodium falciparum and Plasmodium vivax, for example. Same idea on why I haven't worked with, say, South American data to that extent. Yeah.
Interesting, thank you. Any other questions from the floor?
Yeah, I had a question about the difference equation model. Have you also done the model with differential equations, and like how different are the results?
We have not done the full model with differential equations because it's quite large in scope. I think that's a really good question. Obviously we smooth over the dynamics that we're seeing there. And so there are some other differential equation models out there that, or mixed models, I would say. There are some models that look at, say, differential equations to track the immune cells and difference equations to track the parasites dynamic. But there is this, we focused on the difference equation model because of the thought that synchronization was going to be an important aspect. I think that's a really good question that you have, because one of the things that we sort of got out of it was that there is a blurring of these subpopulations in terms of the timing of their presence. I think we would see more of a blurring in the differential equation model, but it may not be so categorically different that what we're working with. I think the other thing that would be important is some sort of, to do that and have sort of a biologically realistic idea, you'd need some sort of cutoff so that different genotypes could actually sort of be lost within the population rather than persist at really low levels and then reappear again. Because one of the things that we don't seem to see is that, the reappearance of certain expression once it's sort of burned itself out, for example. So I think you potentially could, but I think it's a really interesting question. There was a paper that came out a few months ago that compared a number of within host transmission or within host malaria models that has some really neat comparisons about how these different modeling structures play into it. And so I'd be happy to share. It's not one of my papers, but I'm happy to share that reference. I think it's a really great look at sort of how do these different modeling types lead to differences or similarities in their outcomes.
Thank you, Lauren. In the interest of time, we're going to move on to the next talk of today. But interesting talk there, Lauren and Jose and